All right, imagine this. You're a doctor. A patient is rushed in with a really bad, life-threatening infection. You have to act fast, but you have no idea what you're up against. What if I told you there's a test, one that's been around for over 140 years, that can give you a game-changing clue in just a matter of minutes? Well, that's the incredible power of the Gram stain. And today, we're going to break down exactly how it works.
You know, it all boils down to one deceptively simple question. Does the bacteria under the microscope turn purple, or does it turn pink? I know, it sounds almost trivial, right? But believe it or not, that answer can be the difference between life and death. It guides a doctor's next move hours, sometimes even days, before any of the more advanced tests come back. So, how on earth is that possible?
Think of this color test as a fork in the road. It sends us down one of two very different paths, and where we go all depends on how our little bacterial suspects react to what is basically a chemical interrogation.
So on one side, you've got your gram-positive bacteria. These are the ones that hold on tight to a deep purple stain. We're talking about major players like Staphylococcus aureus, you know, the bug that causes staph infections. Then on the other side, you have the gram-negative bacteria, and they end up staining a bright pink or red. This group includes famous bacteria like E. coli. Just this simple color divide, purple versus pink, is the first absolutely crucial piece of the diagnostic puzzle. Now, you might be thinking this is some kind of cutting-edge new technology, but look at that date, 1884. This technique was developed way back then by a Danish physician named Hans Christian Gram. The fact that we are still using it in our modern age of gene sequencing and all that, well, that's a huge testament to just how incredibly effective and brilliant it is. Okay, so to really understand why this color test works, we've got to look at the bacteria themselves. More specifically, we need to look at their defenses. Just think of it as the specific kind of armor these microscopic suspects are wearing. The key ingredient in this armor, the real star of the show, is a substance called peptidoglycan. It's this strong mesh-like material made of sugars and amino acids, and it's what gives the bacterial cell wall all its strength and structure. And here's the secret. The entire Gram stain works because of the amount and the arrangement of this one single substance. And this is where that whole armor analogy really clicks. Gram-positive bacteria, our purple guys, they have a massive, thick wall of peptidoglycan. It's almost like they're wearing a heavy suit of medieval plate mail. But the gram-negative bacteria, the pink ones, their peptidoglycan layer is super thin. Instead, they've got an extra layer of defense on top, a protective outer membrane. It's kind of like wearing a slippery leather jacket over some very thin chainmail. Two totally different defensive strategies. So how do we figure out what kind of armor each bacterium is wearing? Well, we put them through a little four-step chemical interrogation that is specifically designed to make them show their true colors.
This whole process happens in 4 distinct stages, using 4 key chemicals. First, we use a primary stain, a purple one called crystal violet. Second, a mordant, Gram's iodine, which is there to help set the stain. Third, and this is the critical one, a decolorizer, which is usually alcohol. And finally, a pink counterstain, called safranin.
All right, so in step 1, we basically flood the sample with crystal violet, and it just soaks into the cell walls of every single bacterium on the slide, turning them all a deep, uniform purple. At this point, they all look exactly the same. You can't tell them apart at all.
Next up is the iodine. Now this is what's called a mordant. Think of it like a chemical lock. It sneaks inside the cells and it binds with that purple crystal violet. They team up to form this big, chunky complex that's way harder to wash out than the dye was by itself. It effectively locks that purple color in place, for now.
And now we come to it, the most critical moment in this whole interrogation. This is the step that separates our suspects and really forces them to reveal their true identity. This is the whole ballgame right here.
Okay, this is where the magic happens. We add the alcohol, and this is the key that unlocks the whole secret. For the gram-positive bacteria, that alcohol hits their thick, heavy-duty armor and causes it to dehydrate and shrink, basically tightening its grip and trapping those big purple dye complexes inside. The stain is stuck. But for the gram-negative bacteria, the alcohol completely dissolves their slippery outer membrane. This exposes that thin little peptidoglycan layer underneath, which just can't hold on. The purple color gets washed right out. So at this point, our gram-positive bacteria are still sitting there bright purple. But the gram-negative bacteria, they're completely colorless. They're basically invisible under the microscope. So we add the final chemical, the safranin. This pink stain gets soaked up by those colorless gram-negative cells, making them pop out in a nice, bright pink. The purple ones are already stained so dark that the pink dye doesn't even show up on them. Now, of course, this amazing technique really does require some skill. The single most common mistake is over-decolorizing. You leave the alcohol on for just a few seconds too long, and you can accidentally strip the purple stain out of everything, even the gram-positive cells. That gives you a false pink result. Using an old bacterial culture or making your smear on the slide too thick can also mess things up. Precision is absolutely key here.
So let's bring this all back to that hospital bed. Let's talk about that patient. Why does this century-old method of staining bacteria, either pink or purple, remain so completely indispensable in modern medicine?
It's all about speed. In just a few minutes, a doctor gets a massive tactical advantage. Knowing if a bacteria is gram-positive or gram-negative immediately helps them choose the right antibiotic. For instance, penicillin is great at attacking that thick peptidoglycan wall, so it's often effective against gram-positive bacteria. But that same antibiotic would be pretty useless against a gram-negative bug, because that outer membrane just acts like a shield. This test gives the doctor that vital first clue.
And that head start is, well, it's everything. It can take 24 to 72 hours, that's 1 to 3 days, to actually grow the bacteria in a lab and get a definitive ID. In serious infections like sepsis or meningitis, that's time the patient simply does not have. The gram stain is the bridge that gets doctors and patients through that incredibly dangerous gap.